So hello all. Today let's discuss the recent Fmge 2026 question paper. So without any further delay, let's go on with the first question. So the first question here, as you can see, is eruptive xanthomas with chylomicron band on lipoprotein electrophoresis indicates which of the following. So we all know this is something related to hyperlipoproteinemia, right? Hyperlipoproteinemia. So now let's analyze the options given. One is LDL receptor defect. So what type of hyperlipoproteinemia it is? It is type 2 hyperlipoproteinemia. Okay, if you remember, this is type 2 hyperlipoproteinemia, and if to be specific, this is type 2A, and ApoB100 defect comes under type 2B hyperlipoproteinemia. And what do you mean by LPL deficiency? LPL deficiency is nothing but lipoprotein lipase deficiency, which is nothing but this is characteristic telltale enzyme deficient, which is talking about familial chylomicronemia syndrome, which is type 1 to be precise. Okay, this is type 1 hyperlipoproteinemia, which is otherwise called familial chylomicronemia, and broad beta disease is nothing but type 3, which is due to ApoE defect. Okay, this is type 3, which is due to ApoE defect. So here since they've given there is eruptive xanthomas with chylomicron band, so they've given you the in the question itself there's something called chylomicron band on lipoprotein electrophoresis. So the answer would be, yeah, you're right. So the answer would be let me choose. Yeah, the answer would be lipoprotein lipase deficiency, which is a case of familial chylomicronemia. Okay, so this is the answer here. Okay, so now let's move on to the second question. So the second question given here is bicarbonate level is given, which is 14, and carbon dioxide is 24, and pH is 7.3. So they've asked you to interpret the same. So what is the normal level of bicarbonate? So bicarbonate, the normal level is around 24, and what is the normal level of CO2? This is somewhere around 40, right? So in this patient, before going on to this patient, now let me give you a scenario. What happens if, what is the telltale picture if bicarbonate level is going to drop down from 24 downwards? So what is the condition here? If bicarbonate level is decreased, then it is acidosis, right? If which type of acidosis it is? Metabolic or respiratory? It is metabolic acidosis. Okay, so if bicarbonate level is going to be more than 24, then it is going to be alkalosis. Which type of alkalosis it would be? It is metabolic alkalosis. Similarly, if CO2 levels increase, what is it called? If CO2 level increases, if there is CO2 retention, then it is called as acidosis. What type of acidosis is it? This is respiratory acidosis. Okay, and on the other hand, if it decreases, it would be, if PCO2 decreases, it would be respiratory alkalosis. Fine. Yeah, so in this case, we can see that pH level is normal, right? pH is normal. Normal pH level is 7.35 to 7.45. Okay, but then bicarbonate level is less. So if bicarbonate level is less, it is metabolic acidosis. Similarly, your carbon dioxide level is normal level is 40, it is decreased. If carbon dioxide level is decreased, it is respiratory alkalosis, right? It is respiratory alkalosis. So we have two possibilities to explain this picture. So since the pH is not deranged, if pH is deranged, we can simply say that it is either metabolic acidosis or respiratory alkalosis. But since pH is normal here, it means that there is some amount of compensation that has taken place, and that has brought the pH to the normal desired level. Okay, so so there are two possibilities here. Either it could be metabolic acidosis with compensatory respiratory alkalosis, or it could be respiratory alkalosis with compensatory metabolic acidosis. So now let's see the given options. So the two options are respiratory alkalosis and metabolic acidosis. Here it is respiratory acidosis. So it's not a case. Metabolic alkalosis, no, because bicarbonate level is decreased, so it is not alkalosis. Respiratory alkalosis without compensation, again it cannot be a choice because the pH is normal, so this is also not the choice. Metabolic acidosis with compensation, yeah, this could be the reason here because bicarbonate level has gone down and the pH level is also normal, so the answer would be metabolic acidosis with compensation. Fine. Okay, now let's move on to the next question here. So the next question is, a patient with hepatomegaly and intracellular accumulation of lipids and glycoconjugates, which organelle is responsible? Okay, so the key points here is there is hepatomegaly and intracellular accumulation of lipids and glycoconjugates. So basically there is accumulation of glycated substrates. Okay, so the glycation has already happened. Okay, so glycation happens with respect to which organelle? So when you talk about glycation, you're talking about which organelle? Yeah, it is Golgi apparatus. Okay, so Golgi is not given in the option. Okay, if at all Golgi was given in the option, you cannot consider it as Golgi because if the defect is in Golgi apparatus, glycation would not have happened. Okay, but since glycoconjugates are accumulated, it means that they are formed properly. Okay, but they are not able to get destroyed. Okay, they are not able to get destroyed. So where do these substrates get destroyed normally? So anything that is formed from the Golgi apparatus, if it has to get destroyed, it has to go to the suicidal bag, which is called lysosomes. So if there is something wrong with the lysosomes, then these substrates will not get degraded and hence it gets accumulated. Okay, so the answer here would be lysosomes. Now let's move on to the next question here. The next question is, an alcoholic presents with confusion, dizziness, visual disturbances and unsteady gait. So name the vitamin associated. So this is a direct question. I don't think it requires any specific explanation here. So what is a mnemonic for this thing? Goa, what does G stand for? G stands for gait disturbances, O stands for ophthalmoplegia, and A stands for ataxia. So this Goa is associated with what condition? It's called Wernicke's encephalopathy, and that is associated with which vitamin? Yeah, we all know it is thiamine. Okay, so the answer here is thiamine. Okay, so now let's move on to the next question here. So next question is, a child presents with hepatosplenomegaly with histological examination showing foam cells. So foam cells and hepatosplenomegaly. Hepatosplenomegaly simply means there's some accumulation, and there is one more hint that there is foam cells. So what could be the diagnosis? It is a short case of Niemann-Pick's disease. Okay, so it's a case of Niemann-Pick's disease. So what is the product that is accumulating? The product that is accumulating here is sphingomyelin. Okay, and what is deficient here? The enzyme which is deficient is sphingomyelinase. Okay, sphingomyelinase is deficient, so we get sphingomyelin deposits. Okay, so this is it. So if you need, maybe we can have a quick recall of all the sphingolipidoses. So GM1, starting with GM1 gangliosidosis, so that is due to enzyme, the enzyme which is defective here is beta galactosidase. GM2, we have two, that is Tay-Sachs and Sandoff's. So Tay-Sachs deficiency is beta hexosaminidase A. Sandoff's deficiency is both beta hexosaminidase A and B. Fabry's, the defect here is aryl sulfatase, alpha galactosidase. Okay, so Fabry's, it is alpha galactosidase. Metachromatic leukodystrophy is aryl sulfatase A. Krabbe's disease is galactocerebrosidase. Gaucher's disease is glucocerebrosidase. Niemann-Pick's disease is sphingomyelinase. And Farber's disease is ceramidase. So you'll have to remember at least the enzymes and the disorders involved. If possible, the type of accumulations. Like in Gaucher's, we have crumpled tissue paper accumulation and zebra body inclusions that are seen in Niemann-Pick's disease, and cherry red spots and Tay-Sachs. So all those things, if possible, please do remember. Okay, so but for sure you should be knowing this is like a favorite topic with regard to biochemistry. So please do remember all the sphingolipidoses, the diseases, and the enzymes which are defective. Okay, then let's move on to the next question here. So the next question is, a tall person with unusually long limbs suspected to have Marfan syndrome is due to defect in which of the following genes? So elastin, fibrillin, dystrophin, and desmosome. So which is it? So Marfan's, again, this is a direct question, I would say. The answer is fibrillin 1. So why is it fibrillin 1? This fibrillin 1 is associated with formation of what is called microfibrils. So these microfibrils, they are involved in maintaining the architecture of the elastic structures, like the elastic arteries like aorta, and ligaments, many ligaments, and lens, and other things. So since microfibrils are not formed because of defect in fibrillin, these particular structures, they tend to get distorted, resulting in the features of Marfan syndrome. Okay, so with this, let's move on to the next question here. A child presents with self-mutilating behavior. Okay, so this is again another telltale sign. Self-mutilating behavior with hyperuricemia. So these two combines together. Okay, so what is the disorder? The disorder with self-mutilation is always, we know, right? The classical Lesch-Nyhan syndrome, right? Lesch-Nyhan syndrome. So what is the enzyme that is defect here? The enzyme defective is HGPRTase. Okay, so what is the abbreviation? Hypoxanthine guanine phosphoribosyl transferase deficiency. Fine. And is it? So what is just to give you additional points here. So adenosine deaminase deficiency results in, here severe combined immunodeficiency disease or bubble boy syndrome. Let's move on to the next question here. So there is yet another question from sphingolipidosis topic in this paper. So sphingolipidosis, as I told you, sphingolipidosis, glycogen storage disorders, mucopolysaccharidosis. So these three are major domains from which you can expect direct questions. They'll maybe give you the disorder and ask you for the particular type of deposits. Like in this case, they've asked you crumpled tissue paper appearance, and then in the previous case, it was foam cells and Hurler, Hunter. So the prominent ones are like they tend to ask in a form of a direct question, right? So please go through all these things before any of your exams. Okay, now moving on. Crumpled tissue paper like cerebroside accumulation within the macrophage is seen in, we all know it is Gaucher's disease. So Gaucher's disease is associated with which enzyme deficiency? It is beta-glucocerebrosidase. It is beta-glucocerebrosidase. That's the answer. So to reinforce, beta-galactocerebrosidase deficiency is associated with Krabbe's disease. Okay, and aryl sulfatase deficiency is seen in metachromatic leukodystrophy. Okay, metachromatic leukodystrophy. And acid maltase deficiency is seen in your Pompe's, that is not even related to sphingolipidosis, right? So that is glycogen storage disorder type 2. So this is about question 8. So now let's move on to question 9. Okay, so we are beginning with where we left. Okay, so this is a child presenting with cardiomegaly and heart failure. Enzyme deficient is acid maltase. So acid maltase, just now I told you, acid maltase deficiency is associated with Pompe's disease. So one more pointer here is cardiomegaly and heart failure. So the glycogen storage disorder which is associated with cardiomegaly and heart failure is always your Pompe's disease. Okay, so this is it. Okay, to reinforce it again, what is von Gerke's disease due to? It is due to deficiency of glucose 6 phosphatase, right? And Anderson's disease is branching enzyme defect, whereas Cory's disease is debranching enzyme defect. So what is a mnemonic? We have it as A, B, C, D. So Anderson's is for branching and Cory's is for debranching. Fine. This is it. So now let's move on with the 10th question here. A patient presents with ataxia, ichthyosis, retinitis pigmentosa with increased phytanic acid levels. Phytanic acid levels give the likely diagnosis. So they've given you a list of disorders here. So these three are telltale pictures, and again this one, phytanic acid. So all these things point to one condition. What is it? Yeah, it is Refsum's disease, which is due to defect in alpha oxidation happening within peroxisomes, right? Yeah. The other ones are also like adrenoleukodystrophy is also linked to peroxisomal fatty acid oxidation, but this is very long chain fatty acid oxidation. Okay, this is concerned with very long chain fatty acid oxidation. Zellweger's is similar to Refsum's, but then this is more of a neonatal condition. So here, because it's going to be neonatal, we do not consider it. And this thing, Refsum's disease, has this classical picture of ataxia, ichthyosis, and retinitis pigmentosa. Especially, there is something called phytanic acid elevated. Okay, and so what do you mean by alpha oxidation? So basically, in any fatty acids, if you see, there will be beta oxidation, right? But supposing in the beta carbon, if there is a branch point, okay, if there is a branch point at beta carbon, the fatty acid goes in for alpha oxidation, and alpha oxidation happens in peroxisomes. So apart from alpha oxidation, what other types of oxidation happens in peroxisomes? It is very long chain fatty acid oxidation that happens in peroxisomes. What about Eissel's disease, inclusion cell disease? That is due to a lysosomal targeting defect. Okay, a lysosomal protein targeting defect. Okay, so what is the enzyme defect here? Mannose 6-phosphate tag is not there, right? Yeah. Now let's go on to the next question here. The next question is DNA matching technology used for identification of a person. So that is, what is this technology called? This technology is called as DNA fingerprints. Okay, DNA fingerprints. So what do you mean by DNA fingerprinting? So DNA fingerprints is nothing but there are in a person's genome, if you see, there will be about 2 to 6 base pair tandem repeats. Okay, tandem repeats that will be interspersed within the person's genome, and the number of these repeats that would vary from person to person. Okay, and that particular thing, the number of those fingerprints, based upon the number of those fingerprints, we can identify a particular person, a victim. It's also used for paternity testing and other things. Okay, for example, I do have a picture here. So, yeah. Okay, so supposing this is the pattern of the suspect. Okay, sorry, this is the pattern that you got at the crime scene. Okay, so supposing there was some DNA that was somehow collected from the crime scene, and these are the fingerprints. Okay, see, basically what they do in DNA fingerprinting technology is they first extract DNA. Okay, so DNA is first extracted, and then it is PCR amplified. Okay, PCR amplified, and then it is subjected to restriction enzymes. Okay, restriction enzymes, and then we get the fragments are then subjected to electrophoresis. Okay, and based upon the number of these tandem repeats, these fragments, the digested fragments, they vary in size. Okay, supposing the number of repeats are going to be long in one person, then they move to a lesser extent. Okay, so based upon the number of these repeats, the restriction enzyme will cleave. Okay, and post this digestion by restriction endonuclease, we subject them to electrophoresis, and this is the electrophoretic pattern. Okay, this is the electrophoretic pattern of the DNA that was collected from the crime scene, and these are the patterns of 3 suspects. Okay, so let this be suspect 1, this is suspect 2, and this is suspect 3. So which of these 3 suspects DNA fingerprints are matching with that of the one that is bought at the crime scene? So it is suspect 2, right? So suspect 2's fingerprints are matching with the ones that was got from the crime scene. So the so the suspect 2 is the accused, right? So this is how this technique of DNA fingerprinting works. Okay, so the answer here would be, yeah, since they've not given you DNA fingerprinting as such, but what you have instead is short tandem repeats. The closest analogy would be short tandem repeats. Okay, but what do you mean by SNPs? SNPs are nothing but single nucleotide polymorphisms. So this is nothing but a small variation in a nucleotide occurring in more than 1% of the normal population, okay, with no phenotypic or gross morphological change. And DNA mutation is with some clinical features, with some aberrations in the clinical features. Okay, and microRNA, it is RNA, no way related to this DNA matching technology. Okay, so the answer would be short tandem repeats. Okay, the next question here, again a very easy one. I hope most of you would have got this right. Child presenting with bleeding, ecchymosis, no hematrosis, bleeding time elevated, deficiency of which of the following vitamins? So bleeding, ecchymosis, we all know it is vitamin K for sure, right? Why vitamin K? Why vitamin K? Because vitamin K is involved in the synthesis of which of the factors? It is 2, 7, 9 and 10. You're not supposed to draw 2 like this. Okay, 2, 7, 9 and 10. So it is involved in gamma carboxylation of 2, 7, 9, 10 factors, right? So that is why there is bleeding and ecchymosis. Okay. Next thing is next question here. A patient presenting with photosensitivity, purpura and defect in conversion of uroporphyrinogen 3 to coproporphyrinogen 3. So which of the following is involved? So this is again, see, there is nothing to be, even if you forgot the whole pathway, we could have just got it right with using your common sense. So there is something with regard to conversion of one particular substrate to another substrate. So the enzyme should be named after the substrate on which it acts, right? So uroporphyrinogen has to get converted to coproporphyrinogen 3, right? So this should start with, the enzyme should start with uroporphyrinogen. So as simple as that, the answer is uroporphyrinogen decarboxylase, right? So you could have, even if you did not remember the pathway, the porphyrin pathway, you could have simply got this right just by using your common sense. Okay, to reiterate, we'll go on to, so this is the heme synthetic pathway. So we have glycine and succinyl-CoA combining together to form ALA, which is aminolevulinic acid. The enzyme is ALA synthase, and then we have ALA dehydratase from which gives you porphobilinogen. And from porphobilinogen, which is a monopyrrole, we are going to get hydroxymethylbilin, and the enzyme here is porphobilinogen deaminase. And from HMB, non-enzymatic, there is spontaneous conversion of HMB into uroporphyrinogen 1 unless this is acted upon by uroporphyrinogen 3 co-synthase. Okay, co-synthase, if this enzyme comes only, this is getting converted to this, this is actually 3, not 2. Okay, so this gets converted to uroporphyrinogen 3. In the absence of this enzyme, this automatically, non-enzymatically gets converted to uroporphyrinogen 1. Okay, and then uroporphyrinogen 3 gets converted to coproporphyrinogen 3 by the enzyme uroporphyrinogen decarboxylase, and coproporphyrinogen 3 gets converted to protoporphyrinogen by the enzyme coproporphyrinogen oxidase, and protoporphyrinogen 9 gets converted to protoporphyrin 9. The answer, the enzyme is protoporphyrinogen oxidase, and finally we get heme from protoporphyrin, and the enzyme is ferrochelatase. So what are the deficiency disorders associated with each of these enzymes? So ALA synthase deficiency is associated is a disease called, this defect is associated with a disease called X-linked sideroblastic anemia, and ALA dehydratase deficiency, this is called ALAD porphyria. And one more thing that you'll have to remember is lead toxicity affects this enzyme and one more enzyme, which is ferrochelatase. Okay, so lead toxicity can affect two enzymes, one is ALA dehydratase and ferrochelatase. Okay, and porphobilinogen deaminase is acute intermittent porphyria. Uroporphyrinogen 3 co-synthase is congenital erythropoietic coproporphyria. Uroporphyrinogen decarboxylase is porphyria cutanea tarda, and in the case that was given to us, it was the same thing, PCT. Okay, PCT, which is uroporphyrinogen decarboxylase defect. Okay, and CO, coproporphyrinogen oxidase defect, results in hereditary coproporphyria. Protoporphyrinogen oxidases, variegate porphyria, and ferrochelatase deficiency results in erythropoietic protoporphyria. Fine, yeah. So this is what to say, I think this is a series of reactions which you have to be remembering, and the disease again you'll have to remember, know the go. Yeah, and most of it, the enzymes are easy to remember if you remember the sequence of reactions here, because most of the enzymes are named after the substrates on which it acts. Okay, so now let's move on to the next question here. A child presents with seizures. On examination, there is elevated ammonia, glutamine levels, whereas orotic acid level is normal. So this is hyperammonemia. So whenever there is elevated ammonia, there is something wrong with your urea cycle. Okay, so urea cycle, if you can remember, wait, I have the picture here. Yeah, so it begins with carbamoyl phosphate. The synthesis of carbamoyl phosphate, the enzyme here is carbamoyl phosphate synthetase 1. Okay, so if this enzyme is defective, we get type 1 hyperammonemia. Okay, if the next enzyme, so carbamoyl phosphate is getting converted to, getting converted to citrulline by after getting combined with ornithine. Okay, this combines with ornithine and gets converted to citrulline, and the enzyme here is ornithine transcarbamylase, and this defect results in type 2 hyperammonemia. Okay, and the problem is, if ornithine transcarbamylase is defective, this ornithine and carbamoyl phosphate cannot get combined to form citrulline. So what happens to this carbamoyl phosphate? This will get converted to pyrimidine neuropyrin synthesis. So once it goes into your nucleotide synthesis, what happens? It gets converted to orotic acid. Okay, and there is going to be orotic aciduria. Okay, there's going to be orotic aciduria. So type 2 is characterized by orotic aciduria. So the differentiating point from type 1 and type 2 is presence of orotic aciduria in type 2. Okay, and okay, so this citrulline then gets converted to arginosuccinate by the enzyme arginosuccinate synthetase. So if this enzyme is defective, we get citrullinemia type 1. Okay, citrullinemia type 1 is due to ASS defect, and citrullinemia type 2 is due to defect in citrin transporter. Okay, defect in citrin transporter. So because we know this is a reaction which happens in both mitochondria and cytoplasm. So until this, all these things were happening in mitochondria. Post this, it is in cytoplasm. Okay, so this citrin transporter defect results in type 2 citrullinemia. Okay, and arginosuccinate has to then get converted to arginine. The enzyme here is arginosuccinate lyase. Deficiency of this results in arginosuccinate aciduria. Okay, so we get trichorexis nodosa here. And arginine gets converted to ornithine, and the enzyme here is arginase. Okay, arginase deficiency results in hyperargininemia. So this argininemia is characterized by spastic diplegia, if you could remember. Okay, this is characterized by spastic diplegia. This is the key word that you'll have to look for in case of arginase deficiency. And this ornithine has to get combined with carbamoyl phosphate for the further cycle, right? And this ornithine is in cytoplasm. It has to enter into mitochondria, and that is by ornithine transporter. If this ornithine transporter is defective, we get HHH syndrome. So what does this represent? HHH, it is hyperammonemia. Okay, hyperammonemia, hyperornithinemia, and other H is for citrullinuria. Okay, it's not anemia, it is citrullinuria. Okay, so hyperammonemia, hyperornithinemia, and hyperhomocitrullinuria. That is nothing but your HHH syndrome. Fine. So this is it. So in our case, orotic acid levels were normal, but ammonia levels were high. So what type of hyperammonemia it is? Type 1 or type 2? It is type 1. Yeah. Okay. If it were type 2, it would be elevated or orotic acid levels. So this is normal orotic acid level. So the answer would be type 1 hyperammonemia. Okay, this is it. Now let's move on to the next question here. Again, another direct question. Cystic fibrosis, what is the affected channel? So CFTR mutation, we all know it is chloride channel. Okay, so the test that is done for confirmation is your, for confirmation, of course, we have mutation analysis, but the other one is your sweat chloride test, right? A sweat chloride test is the diagnostic hallmark. So it is chloride channels which are getting affected. Fine. So this is it. We have come to the end. So I hope most of you got the answers right. So keep revising. I think it was pretty much an easy paper. It depends on, because most of the questions were direct. Nothing was, conceptual. It was all more or less, it was direct. So keep revising. All the best.